Okay, it's 9 o'clock in Belgium and 15 o'clock in China. Good morning and good afternoon, dear participants, dear professors. We meet each other again in the clouds. Here is the main venue of the third China-Belgium Science and Technology Exchange Symposium, Day 2. So you're sitting in the session 5, Forum of Young Scientists. I'm Chen Kai from ACPB and the host of this session. For session 4, climate change, you might have to switch to another meeting ID, so here. For session 6 and the closing ceremony, you may stay here. The 2 sessions will start later here in the same room. Now let's start our session officially. So Forum of Young Scientists is a podium for the collaboration between Belgian and Chinese universities and the talent development. This session is new this year in the China-Belgium Science and Technology Exchange Symposium because we have realized that the young talents who have strong education and research backgrounds in both Belgium and China will be the core of the scientific collaboration between the two nations in the near future. Therefore, we set up this forum for young scientists and invite them already in the spotlight on the stage. So in addition, we are going to discuss during the roundtable interview on a role model of collaboration between universities and talent development. So there are 2 bullets on today's agenda. The first bullet is the keynote talks by 5 professors, 5 presenters. They are on the screen, Professor Wang Jun, Professor Qian Junbin, Professor Yuan Qiang, Professor Zhang Qilu, and Professor Zhang Zhiyue. They are all rising stars in the research fields covering from biology, medical sciences, civil engineering, chemistry, and the pharmaceutical sciences. So now the first, I'm going to stop. Okay. So now the first keynote speaker is Professor Wang Jun. Professor Wang had studied as a master student in Ghent University around 2009, 2010 till 2012, something like that. Then he went to Germany. He got his PhD in Max Planck Institute for Evolutionary Biology. After he got his PhD, he returned back to Belgium, working as a postdoc by VIB KU Leuven. And then he joined the Institute of Microbiology, Chinese Academy of Sciences in Beijing. So Professor Wang is now a principal investigator with research interest in bioinformatic tools and computational methods for health and disease-related microbiome. So the topics of his talk today is on the screen: the data-driven microbiome research in human health. So Professor Wang, the stage is yours.
So thank you very much, and thanks for this kind introduction. Hello, everyone. It is a pleasure to meet many of you again after a long time of, after a really long while after Ghent and Leuven. And in Beijing, I also have worked for around 5 years on my group. And here are some of our research points that I'd like to share with you and discuss. So the idea of our research is so-called data-driven microbiome. And why do we study microbiome or the microbes in our body? Well, from an evolutionary perspective, the evolutionary history of microbiome actually long succeed or exceeds our own evolutionary history, the world has been, for the majority of the part, microbial. And even after the development or the evolution of eukaryotes, like human, animals, plants, they still participate in large part of our physiological progress and health. For instance, the bacteria in our guts help us to digest the food, keep our immunological system stable, and if something goes wrong or so-called dysbiotic disease would occur. In the last 10 years, it has become a very hot topic in biology because the tool in sequencing and other omics, and we start to learn the importance of those systems. It got so popular that even some of the pop culture, like South Park, picked those things up. And from our own research perspective, why do we care about the health and disease? Well, it really affects our own succession of humankind and the human race. That's why medicine has always been a major research topic or field in biology. So what about our group, or what do we do? We have a very strong focus on methodological improvements or advances, especially under the, well, so sort of the hint or the suggestions from Celine Brainer that usually the new technology brings new data, and then from new data, we start to look at new biological questions and try to answer them. I'm going to give a few examples in the next few minutes. So what we do in our group. So the first is to observe, to understand what is there. So what is in this kind of complex communities, and why do we care about their differences? Well, in the last 10 years, as I mentioned, the microbiome research, as well as many other fields in biology, has been the product or the beneficiary of next generation sequencing technology. For instance, the MySeq I put there, and before that, we also use the 454. But now we have a newer generation, so-called third or the single molecule real-time sequencer, exemplified by nanopore and pachybial. We focus on the usage and application of nanopore in our lab because it's affordable, at least to some labs, and we have the smaller one called Minion that is basically available just next to computer, and their big promission is expensive but still affordable. We did a few things with this new technology, and we utilize its properties, like it's very fast, and you can sequence the whole molecule in one go, and you can detect modifications on those things as well. And from the clinical demand point of view, we worked on a few things. One of the paper we recently published is called the development of so-called MT-NGS or MT-TGS. Basically, we improved from the current available mNGS for pathogen detection, but we replaced the DNA with RNA and cDNA, which has even higher efficiency in terms of the proportion of microbiome to the host, and also it helps us to further reduce time in clinical diagnosis of pathogens. On the other hand, when we are currently still living in the middle of the COVID pandemic, and we now know that this pathogen is actually a virus, RNA virus, and if we put the viral or virus communities into the same level of microbiome, like a bacteria, we have this complex or even more complex communities also in our gut, in our intestines, in the lungs and other parts, which was very hard to characterize because their genomes are small and there's no universal marker. However, with physical enrichment and fast sequencing, we could do this much faster and get a very full overview of this community. Interestingly, after we developed this method, we looked at COVID patients' gut and checked their virome. It turned out if you look at healthy individuals and the patients, their virome are different. And if you look at patients who have they have severe symptoms versus those with slight symptoms or no symptoms at all, their virome are also very distinct. What we learned after using mice model and correlation analysis is that we have quite a lot of these uncharacterized virome members that correlate with the immune parameters of the host and worth investigating. So there might be some causal relationships between the status of virome and the overall status of human immunology. Then these very close collaborations with the clinic and the hospitals, we also help to understand a few mechanistic work or mechanisms in disease development or treatment. One example work was together with the autoimmune department who works on lupus, which is a typical autoimmune disease, and there are many factors participating in it. When we look at gut microbiome in the patients, we realized how there are certain members who are enriched in the patients and other probiotic or healthy members are missing from the gut microbiome. And with a lot of bioinformatic investigations, we could dig into the exact mechanism. So quite a lot of the microbes from the lupus patients actually come from the oral environment, so from our oral microbiome, and they encode a bunch of peptides that can mimic our own proteins, which if expressed in the gut could induce auto antibodies, and that could contribute to the development of lupus as well. Here we first identify the target in microbiome in the gut and trace its origin, and then it also provides a future diagnostic marker or treatment targets for this kind of disease. Another one is sort of a collaboration with traditional Chinese medicine, which incorporates a larger volume of different experience on medicine and the disease they can treat. Here we focus on type 2 diabetes, and one of the old component called berberine. It's a new sort of, it's an old drug with many new findings, and we found berberine as the key component of many Chinese traditional medicine actually induce a significant change in the gut microbiome, and more importantly, it induces improvement or the increase of butyrate producing bacteria, and butyrate is one of the key metabolites that is beneficial for metabolic diseases, including type 2 diabetes. And we didn't stop there, because when we learned from so many needs and so many issues in the clinics, we start to also look at what we can do from our own perspective. That is the work of our third direction, which is utilization. Basically, we try to identify the large reservoir of bioactive molecules using bioinformatic approach, more specifically artificial intelligence. So one of the examples we have as the first success story is we tried to look at the new component called, or a very common component called antimicrobial peptides. Some of them can be transformed or modified into new drugs. And before, this had to be done experimentally because their sequence had no similarity, and there's basically no tool to identify them with high efficiency. But with the neural network, this can be done with very high accuracy, over 90%, and experimentally we can synthesize basically around 200 different peptides, and 180 of them turn out to be active. In overall, we have more than 80% of the success rate. More importantly, a few of them, roughly 10 of them, are very effective against multi-drug resistant gram-negative bacteria, which has had no active drug or new drug in the last 40 years. And a few peptides of ours is very effective even in mouse models. This work is currently in the second round of minor revision at Nature Biotech, and we hope it can get accepted very soon. From there, we even extended our analysis to so-called colitoxin, basically anti-cancer substance in microbiome that has been known for over a century, but it hasn't been systematically studied. With AI and big data, we could actually mine two different kinds of colitoxin. One is peptide, and the other is the microRNA. So far, we were successful in identifying more than 40 different molecules that are super effective in cell models, and we are in the progress of working on the animal test in testing their efficacy in treating cancer. So here is a very short summary and acknowledgement for our work. We have quite a few good collaborators in Europe as well as within China, and we also had quite a lot of different departments funding to get this done. But the central idea is quite simple. We try to connect big data with biology and try to mine useful targets, useful diagnostic markers, as well as products from this kind of huge amount of data. Thank you.
Yeah, thanks a lot, Professor Wang. Your presentation is really impressive, and also many thanks for your good control of time. Before we step into the next speaker, I kindly ask all the audience to switch on your cameras to show the respect to the keynote speakers. And also, meanwhile, if you have any questions, you can just type your questions in the chat box, so the presenters can answer you in the chat box as well. Due to for the sake of time, we are not going to have the online Q&A steps. So the next keynote speaker is Professor Qian Junbin. Professor Qian started from 2009 to 2014 as a PhD candidate in the Department of Molecular Cell Biology in China-Belgium. And then he joined the Zhejiang University and became a research professor, and now he's a doctoral supervisor as well. Professor Qian has expertise in the studying of the pathogenesis of critical diseases, developing the early diagnosis biomarkers, strategy for the clinical trial-based treatment stratification by single-cell multi-omics sequencing. So now it's all yours, Professor Qian, please.
Hi, everyone. Thanks for the kind introduction, and it's my pleasure actually to introduce about the single-cell technology. Actually, I'm a PhD and a postdoc in KU Leuven VIB, and just went back to China to start my own group last year. So everything is still fresh here. Here I want to introduce you single-cell technology because it's a really great innovation. And here I give you an analogy. It's actually from our fruit plate. So traditionally we just blend it and then goes to the juice, and then we just taste it. The very vague of idea of what's inside, but then it's more than art than the science. But now with this technology innovation, we can really sort this fruit into different categories, and then find out really which one actually is in problem, and then this problem can cause other problems to other types of fruits. So basically it becomes a classic physics upgraded to atomic physics or even relativity. So to me, it's actually a kind of revolution in the increase of dimensionality. So this technology is being excuse me, it's not moving. Okay, now it's going. Okay, this technology is advancing actually rapidly every year. From 2009, it's really a single cell. We can only assay one cell at a time with our mouse tube, but now it's been scaling up every year. So now actually we can assay 1 million cell at a time. Even we can incorporate more dimension, like the spatial dimension is also incorporated into the single cell sequencing field. You can see that more and more papers published every year, and that's really a booming technology. So the idea here is simple, that you can take out your tissue, our liquid biopsy, and then get into this single cell suspension. And then for each cell types, you can assay different modality, or so-called multi-omics. So most of the time we can assay this RNA expression, but actually in the same time, you can concomitantly also look at DNA methylation, chromatin accessibility. For instance, this EC site-seq technology can assay in the same time 4 different modalities. So it's really powerful tool, and its application is not limited to basic research from animal to IPS to organoids, but actually more interesting, or at least we are more interested in the disease. So by comparing the healthy and the disease tissue, we want to actually look into the mechanism of this pathology and to have new diagnosis and new target for treatment. Today, I will just give you several examples to show how to use single cell sequencing technology to do research from basic perspective and then translate into the clinical interpretation. The first one is actually the COVID-19. We know that actually the virus can infect our lung, and then our host have the viral response, but then in the same time, it will accumulate inflammatory response eventually. For most people, it will keep some mild symptom, eventually recover, but to certain percentage of people, they goes to critical status and even die. But then we don't know what's the critical part to how to say, to discriminate these two fate. And to do this, we actually profile the immune landscape of this lung fluid. Basically, we take first the lung fluid from the patient and then pass this to the 10x genomic single cell platform. And we assay like 3 modality, which is the RNA itself and TCR sequence and BCR sequence. So single cell is very good at classify different cell types or do the annotations. Here, as you can see from this figure, each small dots actually represent one single cell. When these dots are very close to the other one, and they are belonging to same cell type. So we can here see at least 18 different cell types. So we can do a quantitative comparison if certain cell types is more abundant in certain physiological or pathological condition. For instance, the COVID-19 patient, they have very high level of neutrophils in their lung fluid, and but there's low alveolar macrophage. When you compare the mild and critical disease, you see that alveolar macrophage is still very low in the critical disease, suggesting this is a key pathological aspect. And we look at not only the quantitative data, but also looking at the qualitative changes. For instance, what changed if the COVID patient happened and their immune system is responsive to that. So here we just give one example for CD8 T cells. We can actually monitor their differentiation status. So for instance, CD8 T cell can differentiate into 3 lineages: the CD8 TRM, CD8 TMRA, and the CD8 TX. They are different lineage, and each lineage actually is with different functions. For instance, for the TRM lineage, we can see that, so for this purple color is a mild disease, and the critical disease is the yellow one. So TRM, you have better differentiation in TRM for the mild patient, but for the critical patient, you have higher differentiation for the TEX lineage, suggesting TRM actually is a protective cell type or differentiation lineage, while TX is exacerbates the disease severity. And when we look at the TCR level, because when the T cell expand, their TCR richness will decrease. And when we look here, actually we see the TRM. In the mild disease, they have huge decrease in the TCR richness, and suggesting the antigen-specific TCR actually increase and expand, which is protective. Okay, and then we also see a mini signature that is changed or dampened, especially for the interferon response. For these three major type of immune cells, the critical disease have much lower level of interferon response. And we don't really know the reason, but we also in the same time see that the plasma cell, which express antibodies, is much more abundant in the critical disease. But that's strange because normally antibodies protect us against the COVID. And now if you have more critical disease, you have more antibodies. So some antibody must be bad. And then in later two studies, they showed two mechanisms that is describing these two bad antibodies. First one, actually some antibody can cover the surface of immune cell and then stop the interferon response signaling. And another type of bad antibody is actually the autoantibody against type 1 interferon. So in actually 10% of such severe patients, they develop this autoantibody to block its own interferon. So that's why the interferon response is lower. And we can not only look at the RNA, but also the RNA of the cell itself, but also look at the viral RNA, because some cell, either they are infected by the virus, or some cell actually they eat virus and then to clean virus, for instance, immune cells. Indeed, using an existing tool, actually we develop a little bit more to accommodate our new purpose. For instance, we look at the SARS-CoV-2 counts. We see actually not only the epithelial cell having the viral counts, but also the immune cell like myeloid lymphocyte. They also have the viral counts. And then we, for the first time, look at the differential expression of 11 viral genes and look at their abundance. We see that this S gene is more abundant in the epithelial cells, and the N gene and OFF10 gene are more abundant in the lymphocyte and the myeloid cells. So just in this different gene have different mechanisms for the pathological process. So when we look at here, for instance, the epithelial cells, when they are expressing S genes and their interferon response is being dampened. And that makes sense because virus will actually stop the propagation of the epithelial cells. And for neutrophil cells, when they are having this N, and definitely they will have more interferon response. So in the same time, we can also look at the microbiome. That's the field of Professor Wang. Indeed, we collaborate with his former supervisor, Professor Jeroen Rues, and we found that actually for the bacteria, the monocyte-derived macrophage and neutrophil, they are having more bacteria reads. And indeed, we found that the COVID-19 patient have a distant spectrum of bacteria in their lung fluid. And more importantly, we look at the bacteria-associated cell, and we see that when those cells are associated with bacteria, they are having more pro-inflammatory signals, suggesting this bacterial infection can also contribute to the disease severity. And finally, we can use single cell actually to identify therapeutic targets. For instance, the widely used disomethaone that's actually used for the critical disease, and we found indeed they are a very good target because it target at least 2 groups of cells in the same time. Of course, there's other target that's in clinical test or not found, and in our study actually give new perspective on that. Okay, secondly, I want to talk about the pan-cancer studies. So normally you know that tumor is actually not only the cancer cell, but also the cell surrounding it. There's many types of the stromal cells. So what's the difference between these cell types and how different they're, or how similar they are, and what's the clinical implications? So you can look at all different spectrum, but we selected 4 cell types to start with as our pan-cancer study. So basically we have the lung tumor, colorectal tumor, and the ovarian tumor, also breast tumor to start with. And then we build a stromal blueprint for at least 68 type of stromal cell types. And we found among them 46 is shared and 22 is tumor specific. Okay, and what's the use of that? Of course, that's very good for the basic research to annotate cell types. We use it for also immunotherapy interpretation. For instance, we take the melanoma data and we predict at least there's 2 other T cell population can predict the melanoma patient, whether they are responsive or not responsive to immunotherapy. In the same time, we can also predict the drug response for different tumors. For instance, this CD8 differentiatory pathway, you can see that this LC is lung. So lung has a very good differentiation in this lineage, and ovarian has very low differentiation. And indeed, from the clinical data, we know that the PD-1 treatment for lung is super, and for ovarian, most of the clinical trial failed. Okay, then we really go to the clinical trial, and we want to study whether this PD-1 treatment will work for breast cancer or not. If it doesn't work, and why, we want to answer such question, and also want to know the mechanism behind it. So PD-1 and PD-L1 treatment, that's something we take the approach to look at their early response, because we actually start with early breast cancer patient. We take a needle biopsy, we sequence them with single cell, and then we give them 9 days of anti-PD-1 treatment. After that treatment, we resect the tumor, and then again, we also sequence the single cell. And then we compare before treatment and after treatment, and within these 9 days, what happens. So first of all, we want to know the drug target. Normally, the T cell express PD-1 and other immune cell express PD-L1, or cancer cell express PD-L1. And we found that indeed the PD-1 is expressed in 2 cell types of T, and that's CD4 TEX and CD8 TEX. And indeed, their abundance really associated with responder and non-responder. But surprisingly,
we are a little bit behind the timeline, so you still have half a minute.
Okay, I will just go through this. So we also look at the mechanism of the response to identify more genes. Of course, finally, we want to find the biomarker for patient stratification. And actually, single cell is now already being used in directly making decisions for the patient. For instance, in this drug-induced hypersensitivity syndrome, the clinician can really use single cell to identify a target. And after treatment, you can see that the patient recovered well. Otherwise, this patient just wait to die. So I think in the near future, single cell will be directly involving in the clinical decision, and we are working on that. Finally, I would like to acknowledge my team, and especially my collaboration in Leuven, especially my supervisor, Dieter Lambert. Thank you.
Okay, thanks a lot for a really informative presentation. Due to the timeline, I will step immediately to next speaker, Professor Yuan Qiang, who graduated and got his PhD from Ghent University in 2008. He's now a full professor in the Central South University. Professor Yuan's research interest includes the 3D printing concrete, biological behavior concrete, so cement-based materials, and also durability of the concrete. So it's really, he has a lot of concrete interest in concrete. So now, please, Professor Yuan, for your presentation. We cannot hear you, Professor Yuan.
Yeah, sorry about that. Can you see my screen?
No problem.
Okay, that's good. Good afternoon, everyone. I'm Qiang Yuan from Central South University. I'm honored to be here, and thank you, Chen Kai, for your invitation. I obtained my PhD degree in 2009 from Ghent University. That's 12 years ago, quite a long time ago. And my topic is cement-based materials for slab track of high-speed railway. I would like to first fill you in on some information about the development of high-speed rail in China. Then I'm going to talk about cement-based materials for slab track, and specifically, I'm going to focus on the China rail track system type 3 slab track and self-compacting concrete applied in this track. First part, background. 15 years ago, there was no high-speed rail in China. I still remember 2006, I was first in Europe. I was shocked by the TGV, the high-speed rail in Europe. And at that time, I was thinking, when my country will have high-speed rail line. 15 years later, the high-speed rail network is all over the country, and most big cities are connected by high-speed rail with running speed of 300 to 350 kilometers per hour. And in China, you know, high-speed rail becomes the first transportation choice for people traveling. After 15 years, China has the biggest high-speed rail network and account for 65% to 50% of the world total. And Chinese government like to make ambitious plan, always 5 years plan, and trillions of dollars will be invested in high-speed rail. At that time, almost all big cities will be connected by high-speed rail. And basically, there are two types of high-speed rail technologies. One is real rail technology and magnetic levitation technology. Speaking of maglev technology, the first technology is in Shanghai. It's originated from Germany. Probably it's not that successful. Probably will be demolished. And it's said Japan will build a new high-speed rail maglev line and will open on 2027. And in Changsha, China, right here in my city, a low moderate speed maglev line already in commercial use. And today, my topic will be real rail technology. And this is the most used one all over the world. Traditionally, railway is built on many aggregates. We call it ballast. Like in Belgium and in France, they build a high-speed rail on ballast track. And they hit the running speed record of rail at 570 kilometers per hour. You know, in China, the highest running speed is, guess what? It's 486 kilometers per hour. So it's still much lower than that. However, most countries, including Japan, Germany, China, we all use ballastless track. And the ballastless track has been widely implemented all over the world, despite high initial investment and noise problems. It has obvious advantages over ballast track system, such as increase capacity and reduce maintenance and life cycle cost, reduce the number of track maintenance operations, and thereby increase safety. Okay, part 2 is about the cement-based materials. And Chinese company has developed 3 types of ballastless slab track. CRTS 1, 2, 3 slab track. CRTS 1 originated from Japan and has been used in China for over 2,000 kilometers. And CRTS 2 originated from Germany and has been used for more than 4,000 kilometers. And CRTS 3 is originated from China and will be used for the rest of the new lines in China. Probably 20,000 kilometers, I don't know. And to your left is CRTS 1 slab track, and to your right is CRTS 2 slab track, and the bottom is CRTS 3. They are quite different in terms of structure, but the structure is not our topic today. We're going to talk about the major cement-based materials used in these structures. There are some special cement-based materials used in these structures. Look at this structure. On the top is the prefabricated steam-cured concrete slab. And there is a cushion layer for slab tracks. For CRTS 1 slab track, a mortar with low elastic modulus is used as a cushion layer. And the CA mortar with high elastic modulus is used for CRTS 2 type slab track. And CRTS 3 slab track, we don't use any asphalt. We just use self-compacting concrete as cushion layer. And CA mortar is a kind of asphalt modified cement and made up of cement, emulsified asphalt, water, sand, and some chemical admixtures. CITS 1 and 2 CA mortar are different from each other in terms of compositions and properties. They use different types of emulsified asphalt. The two important parameters are asphalt to cement ratio and water to cement ratio. Therefore, the mechanical properties and durability are different. You can see the compressive strength of CRTS 1 CA mortar is very low, just 1.8 MPa. And due to asphalt cost is much higher than cement, and CRTS 1 CA mortar is more expensive than that of CRTS 2 CA mortar. And CA mortar is mixed by a customized mixing truck, so we can mix CA mortar along the high speed line and cast it immediately after mixing. And here I am. You can see that's like 10 years ago on the job site. Behind me and under my feet are high-speed rail lines. Yeah, I was at that time I was responsible for the consultancy of CA mortar. My duty is to help engineers to cast the CA mortar correctly and solve all the material related problems on the job site. And steam-cured concrete is not a new technology. In the construction of high-speed rail, many structural elements are prefabricated in factory. And steam curing is used for accelerating the production of elements for two reasons. One, better quality control. We build all the elements in factory so we can control the quality quite well. And faster building speed. The 1 day strength with steam curing can reach 14-day strength in normal curing. And this is the curing regime of concrete. So the curing temperature reaches around 60 degrees. And you can see this is steam cured concrete. The slab is about 7 to 9 tons. And the box beam, you can see a big stuff is about 900 tons, are all made with steam cured concrete. This partly explains why China built high-speed rail lines so fast. Normally, for a several hundred kilometer line, it takes 7 to 5 years from design to complete. Okay, now I'm gonna go to a more specific material. And this is the structure illustration of CRTS-3 slab track. From top to bottom are prefabricated slab, SCC layer, base concrete plate, and SCC is cast into the very thin chamber between slab and base plate, and SCC flows on the geotextile instead of concrete. Probably you will say it's just a normal concrete, not special at all. Let me explain a little bit. For conventional SCC, the upper surface is open to atmosphere and a slight bleeding or bubble floating is allowed. It can be repaired by finishing process. However, in the case of sealed space filling concrete, strong bonding between the upper surface and above slab is required, and the bleeding and bubble floating is not allowed because they impair the interface bonding. What's more, the space is very narrow, flat, and quite big. This makes it more difficult to cast. In addition, concrete flows on flexible geotextile and with heavily steel reinforcement. This increases the flow resistance of SCC. And in terms of the structure requirement, the property of hardened SCC were proposed. The compressive strength, elastic modulus, a comprehensive laboratory and full-scale field test was carried out in my lab. And I'm not going to discuss specific tests, just some important general ideas. We designed some special testing setup to simulate the flow of SCC in theater space. A test setup like this. We use the concrete equivalent motor in the test. After casting, check the surface quality by image analysis. For example, there is a big void in the picture, but the other area is good. Obviously, the air is entrapped during casting. We should improve casting method to avoid entrapped air. And this picture is full of small bubbles, and the bubbles may come from the air content of concrete. After casting, the air bubble rise up to the surface. In this case, we need to improve the mix design or raw materials of concrete. We also conducted lots of full-scale tests to check the surface quality and the interface bonding of SCC. There are many failure examples. Look, the interface bonding is terrible, and bleeding segregation you can see here.
Sorry, Professor Yuan, to interrupt you. You have half a minute because we are already behind the time.
Okay, yeah, thank you. Yeah, and after all the work, we proposed the specification for the concrete and all the design principle of SCC. And this is the typical mixture design of SCC. And this is how we build the CRTS3 ballast slab track, and how we cast concrete in the job site. And this is the job site when we do the full scale test. And the technology developed by Central South University has been applied in high-speed rail of 1,647 kilometers. The number is still increasing. So that's all. Thank you.
Thank you, Professor Yuan.
Thank you very much.
Yeah, so we step into the next speaker, Professor Zhang Qilu. He did his PhD studies in chemistry from 2010 to 2014 in Ghent University. And then he started postdoc in the University of Helsinki before he joined the Jiaotong University. Now he's a PI, focused on design, synthesis, and the properties of soft nanoparticles, as well as the potential applications in energy and the biomedicine. So please, Professor Zhang, to welcome you to the stage. So you have 12 minutes and you will hear a notification at minutes 10.
Right, thank you, Kay, for your very kind introduction. So my name is Qilu Zhang. I graduated from Ghent University in Department of Chemistry, Organic Chemistry, and Macromolecular Chemistry. Now working in Jiaotong University. So basically, I'm now working mainly on functional polymers based on nano-sized soft particles. So here I actually will, in this section, I will show you a little bit of work that I have recently done in my group. So the inspiration of this work is the proton functionality and the proton structure. As we have seen this picture, or as we all know, that protons always contains varieties of subdomains to serve for different functionality. As a polymer chemist, a lot of people are trying to mimic the structure and the functionality of protons. So we know that for protons, there are only about 20 monomers that can have a lot of functionalities, but for polymers, we actually have numerous of monomers, but only with very limited functionality. So when we study the structure of proton, we find out that usually there are a lot of subdomains that is fixed in a preformed structure. For example, for example, in this hemoglobin, that you have some alpha subunits that can have the catalytic effect. So in this case, scientists, especially polymer scientists, are trying to mimic the multifunctional structure of protein to use, for example, a tri-block copolymer, as you see on the left side of the screen. And then you have a tri-block copolymer with a fluoride block, a aliphatic block, and a hydrophilic, or we also call it a soluble block in water. So when you throw this polymer in a good way in water, then it forms a kind of so-called multi-compartment nanoparticles. Or in another way, you can have a linear tri-block copolymer, and you have A, B, C blocks with different solubility, and then you allowed it to self-assemble in a no solvent for B block, and then you form a micelles. Then you change the condition of the solvent, then you're able to have make some kind of a different shape of blocks. Then you can further self-assembly into multi-compartment nanoparticles. Those techniques looks very good, but usually if you have a tri-block copolymer, then usually the result structure of multi-compartment nanoparticles are not so stable. Then it tends to be dissolved again when you dilute it, or when the polymer nanoparticle has been forced, undergo some force. So what we are trying to do, we try to develop a new way that we can mimic the structure of a protein. So we go back to the self-assembling of protein. So for example, usually a protein has a primary structure, what we call it polypeptide, and then it self-assembling to secondary structure like a beta sheet or alpha helix. Then further, it forms a tertiary structure. This tertiary structure already have some functionality, and then this tertiary structure finally form quaternary structure, real protein. So when we find out this, we are trying to mimic. We wonder that if we can already prepare nanoparticles or prepare a polymer that can directly mimic the tertiary structure. We jump the first and second structure, directly go to a tertiary structure. So we find out that a star polymer with a lot of arms can serve as these functions. So for example, in this case, the blue part here can be a hydrophobic part, which can serve as some functionality, and the outside can be hydrophilic or water soluble, can protect this structure, for example, in water. And then we attach a very long polymer chain, then you can have an amphiphilic structure of this polymer, and then we allow it to self-assembly into maybe some kind of multi-compartment nanoparticles. So here I will report two examples. The first is a multi-compartment nanoparticles with hydrophilic domains. Usually, if we have polymer self-assembly, you can hardly have hydrophilic domains in the polymer, in the result self-assembly structure, because the hydrophilic part usually migrates outside of to form the shell of the particles in water. So here we design a polymer, as you see on the right side. This polymer structure has this blue part polyacylic acid, and the outside we have polystyrene to protect this hydrophilic part, and then a linear polyethylene glycol to introduce some to induce the self-assembly. Then we have three-step. We use a three-step polymerization and modification, and we have a series of polymers. Those are very well characterized, and then we allowed it to self-assembly in water. Basically, we first dissolve this polymer in an organic solvent, and then we slowly add water inside this solution, and based on the structure of the polymer, we able to form, for example, a multi-compartment micelles, which you see on this screen, the model of the nanoparticles. Here you have the blue part as a hydrophilic part, and the yellow part as a hydrophobic part. In this case, we can already call it a multi-compartment nanoparticles. While tuning the structure of the polymer, we were also able to have to make the hollow structures, which we call the multi-compartment vesicles, and we also have hydrophilic domains and hydrophobic domains. And then we also able to form a string shape of the multi-compartment nanoparticles with alternatively the blue part and the yellow part. And the second work is the crystalline multi-compartment nanoparticles for drug delivery. Why we want to do this? Because in the previous work, we found out that such polymer structures are not so stable in water against dilution. So we actually design a polymer with a crystalline polylactic acid to allow it crystallize to form a very stable nanoparticles. And we hope that such particles can be very stable against the dilution and against the strain shearing, so that it might be able to deliver some drugs for application. So so we first prepare a lot of polymers, and then we allow it to self-assembly into various structures like plates or spheres. So we then we try to load model molecules like Nile red in the plates or spheres, and we find that the loading is quite successful. And with then collaboration with Professor Zhiyue Zhang, who will present later, we are able to allow this Nile red loaded nanoparticles into a cell, into a cancer cell. Then we can see from this picture, we can see that such particles can be successfully loaded in the cell. So to conclude, we actually have been inspired by the self-assembling of protein, that we are able to make a series of multi-compartment nanoparticles. Those nanoparticles have different subdomains, and in the next stage, we are trying to use those nanoparticles to deliver some drugs to allow it to release in body. Finally, thank you very much for your attention. Thank you.
Thanks a lot, Qilu, for your interesting talk. It's a pity that we don't have time to discuss. So the next and the last keynote speaker is Professor Zhang Zhiyue. He obtained his PhD in pharmaceutical sciences from Ghent University in 2016. And then he did also his postdoc in the same group. He joined Shandong University, and now he's mainly engaged in the design and development of pharmaceutical carrier materials and the multifunctional nano drug delivery system. So please, Professor Zhang, to present your talk.
Okay, thanks a lot for the invitation and the introduction. Hello everyone, I'm Zhiyue Zhang from Shandong University at the Department of Pharmaceutics. Today, my topic is raft polymers for drug delivery system. So, just want to do a simple introduction. I graduated from Shandong University at 2012, and then went to Belgium to get the PhD degree from the 2012 and to 2016. And then after 2 years of postdoc training, I went back to Shandong University as a professor. And now my research interest includes includes 3 parts. One is novel drug carriers. Second one is nanoparticular drug delivery system, and the final one is the combination anti-tumor therapy. Yes. Okay. So now I focus on the design of novel polymers, and then use the polymers for the drug delivery system, and then for the anti-tumor combination therapy. So this is my research group, and because I just went back to China 3 years, so my group is still quite small. So the polymers for drug delivery actually has been developed in for more than 6 years. But the first paper reported by Rob Longer for the polymers for the sustained release of proteins and other macromolecules in 1976. After that, the polymers for the drug delivery has been developed very well. So today I want to introduce the raft polymerization. Raft polymerization just use monomer and the chain transfer agent to do the polymerization. And then because the polymer has a sulfur end group, so it can bind to the gold nanoparticle surface by the gold sulfur containing. So today the contents for the topics for the today is including 3 parts. First one is the engineering the interaction between polymer-coated gold nanoparticles and the living cells. So the cellular uptake of gold nanoparticles can be influenced by the size, shape, and the surface chemistry of gold nanoparticles. But then the hydrophobicity of polymer coating, whether it includes the cellular uptake of gold nanoparticles, is not investigated. So we just developed several series of polymers, including different ratios of hydrophobic here monomer and the hydrophobic mere. So with increasing the ratio of mere, the polymers become very, very hydrophobic. So then the gold nanoparticles was synthesized by the reduction of gold salt, and then the polymers was anchored on the surface by ligand exchange. So to investigate the cellular uptake of gold nanoparticles, we developed a new flow cytometric dating strategy to quantify the gold nanoparticles inside the cells. So we can see this with increasing the concentration of gold nanoparticles, more gold nanoparticles can be uptaken by the cells. So we just want to investigate the hydrophobicity of coating of gold nanoparticles can influence the cellular uptake. So we can see this result with increasing the hydrophobicity of polymer coating, more nanoparticles can be uptaken by the cells. So we are not sure whether our results are successful. So we will use another method we call ACPB mass to quantify the amount of gold nanoparticles inside. So the results are similar. So we can conclude that the FACS strategy can be successful in determining the cellular uptake of gold nanoparticles. The TM imaging also confirms the more hydrophobic coating, the more gold nanoparticles can be uptaken. So the first conclusion is the hydrophobicity of polymer coating of gold nanoparticles determines the extent of cellular uptake of polymer-coated nanoparticles. The second topic is engineering the interaction between polymer-coated gold nanoparticles and the influenza virus. So the green one is the TM imaging of influenza virus, and the surrounding is the gold nanoparticles. They also use the same method actually, because the influenza virus has a hemagglutinin on the surface. and the hemagglutinin can bind to the sialic acid on the surface of like red blood cells. So we just want to develop a gonad particles with sialic acid on the surface to determine to investigate the interaction. So the first part is the polymer synthesis. We also use the rapid polymerization to polymerize the HPMA and APMA, because APMA has an amino group which can bind to the lactose by the reduction amination. So in this case, the sialic acid group can bind to the polymers. We use this to alpha-2,6, and also the polymers were anchored on the surface of gonad particle, like the previous topic. And first, we investigated the gonad particle in interaction with lectins, because we use the alpha-2,6 sialic lactose, and the 2,6 sialic lactose can bind to the SNA specifically, but the 2,3 sialic lactose cannot bind to the MAA. So we just first do the DLS analysis and to check the size increase of gonad particles. In this case, if the gonad particle bind to the SNA, the increase of gonad particles can increase. The size of gonad particle increase. So the control, the nanoparticle cannot increase with the MAA. So next, we tested the gonad particles interaction with virus, because the gonad particles have a size of like 50 nanometers, but the virus has 200 nanometers. If we mix them together, we found two peaks. The first one should be the suspended gonad particle, and second one we think that it could be the binding of the gonad particles and the virus. So we also checked the TM and found that the gonad particle was anchored on the surface of the virus. The control the glycan polymers without sialic acid cannot bind to the virus. So the gonad particle functionalized with polymers containing sialic acid chain and can bind to the surface of a virus specifically. The second topic is the design of novel thermosensitive rapid polymerization. We know the LC-acid polymer, which has a lower critical solution temperature. That means if the temperature is above the LC-acid, the polymeric nanoparticles collapse with a partially released drug. As a control, if we use the UCST polymer as for the drug carriers, if the temperature is increased, is above the UCST, the polymer nanoparticles can disassemble with the completely released drug. But normally, actually the UCST polymer cannot be used in the carrier or the drug delivery, because some UCST polymers are too sensitive to the polymer chains, to the ions, and also some polymer has UCST behaviors only exhibits the UCST in the mixture of like solvent and with the DMSO, also the water. So in this case we investigated we want to design UCST polymers that can be sensitive to the physical conditions. Like in this case, the polymers can be reversibly can show the reversible UCST behavior, and then can be hydrolyzed to the previous polymers. This is the monomer synthesis route, because we use the monomer or HPMA, and then HPMA was functionalized with the GE, and then the monomer was polymerized by raft. Okay, this is the final polymers, and because the polymer has a carbonate group, it can be hydrolyzed to the poly-HPMA. The poly-HPMA is it's frequently used in the drug delivery care system. So we investigated the thermosensitive. So because from this picture, we found that at 60 degrees, the polymer solution is transparent. But when we cool down the temperature to 25 degrees, the polymer solution became turbid. And then after heating cooling cycles, the polymers has the thermosensitive properties. And then we checked the different characterizations, and I found that at pH 7.4, the polymer still has the UCIT behaviors.
So we have 1 minute left. We have 1 minute left.
Okay. Okay. Next, we checked the LCST polymers for encapsulation of hydrophilic BSA phase and hydrophobic non-red, and found that only hydrophobic non-red can be encapsulated into the polymers. But for the UCIT polymer, the polymer can encapsulate hydrophilic phase BSA and hydrophobic non-red. So we also do it in the animal, in the mouse, and we found that the phase BSA can be released suspendedly. So the conclusion 3 is the synthetic, transparent, transitionally thermoresponsive home polymer with UCIT behavior within a physiological relevant windows was designed. So to include the raft polymers can be a great interactive materials which can interact with living cells and the influenza virus in an interesting way, and can be excellent drug delivery carriers. Okay, thank you so much, and welcome to China University.
Thanks a lot, Zhiyue. Although we have really limited time for the keynote speakers, but let's give a round of applause to all the 5 keynote speakers for their informative and impressive presentations. Now we're switching to the second bullet. We have invited 3 guests to the roundtable interview. Professor Desmet is a full professor in the Faculty of Pharmaceutical Sciences at Ghent University, member of the Belgian Royal Academy of Medicine, member of the European Academy of Sciences. Professor Huang started pharmaceutical sciences in Professor Desmet's group and got his PhD in 2011. And then he did his postdoc at the Swiss Federal Institute of Technology, Lausanne. Since 2013, he became a professor in the Nanjing Forestry University. Professor Xiong obtained his PhD in the same group in 2017. And now he is also a professor at the Nanjing Forestry University. So the 3 professors have built up and been supervising a joint laboratory of advanced biomedical materials between the Nanjing Forestry University and Ghent University. For years, the laboratory has been fruitful in both scientific research and talent development. So now we are inviting Professor Desmet to give us a short introduction to the laboratory. Please.
Thank you. Good morning. I'll try to share my screen first.
Yeah.
Can you see it?
Yes.
Okay.
Yes, we can.
That's fine. Okay. So thank you very much for your kind invitation to shortly introduce you to our joint laboratory. So it's a joint lab between the Nanjing Forest University in Nanjing and the University of Ghent. So let me first tell you a bit about the beginning of this lab. And actually the very beginning is, let's call it the official start, which was around spring 2019, when a delegation from the Nanjing University actually visited us in Ghent in 2019. And then some months later, actually, we had the real official start when the vice rector of Ghent University actually joined us in Nanjing and signed this kind of official collaboration between Nanjing Forest University and Ghent. So this is the official start, but let me first tell you a bit more about the real start. And in fact, the real start of this joint lab, it goes back almost 15 years ago, when a very young student contacted me and asked me whether he could join me as a PhD student in Ghent. And of course, this was Qiaobo Wang, who is now the professor in the Nanjing Forest University. And actually, you know, since 2007, so Qiaobo, of course, started to study with us. And, you know, we became good friends, so to say. As you can see here, he's in my home on the occasion of Christmas in 2007, I guess, a few months after he arrived in Belgium. So Qiaobo, he also became the president of the Chinese Students Association in Ghent University. Here you see him together with the rector of the university and also together with the ambassador of China in Belgium. And at the right side, you have, you see Professor Qiaobo Wang on the occasion of his doctoral defense in 2011 in Ghent. And important is that after he finished his doctoral defense in 2011, he moved to Switzerland. But as you can see, we kept our relations. So we stayed in contact, because often when people leave the lab after PhD, you lose contact. But the point is that we kept contacts. So Qiaobo, he came back in Ghent in 2015 on the occasion of celebration. I went to visit him in China. And even you can see in 2018, so he got his third child and his name was Paul, given Paul after, and his name was given, the name of Paul was given after, let's say, the name of my brother, who actually died the year before. So just to tell you how close our friendship is and how close our collaboration is. So why do I tell you this little story? Because in the end, you have to realize that such a kind of joint lab as we have it today, it goes back to a young person having an interest in a professor in 2007, in the work of a professor on the one hand, and a professor being interested in a young student, like Qiaobo in 2007. So it's about having a kind of common interest, about willing to work together, willing to start to work together, and willing to walk together. And so that's the real origin of our joint laboratory 50 years ago. So I will tell you a little bit about the current status to give you a short idea on this. And so this is our lab in Nanjing. So it's a 500 meter squared lab. I even have an office there. Of course, it's a long time. I have been in my office now in Nanjing. So this is more or less the group picture. It's a group picture of, I guess, 2 years ago. But we have, of course, a number of PhD students and master students in the lab and postdocs. So important as well, or an important milestone as well, is the fact that, you know, at the right side of me, you can see Dr. Huaqiong, so Ran Huaqiong. So he recently joined the joint lab in January 2021 as a new professor, which is an important milestone for our joint lab. And so let me just introduce you to Ran Hua. So he joined our lab in Ghent as a PhD student. And actually, after that, he indeed realized to get a postdoc scholarship from the FWO, which is the most prestigious scholarship for postdoctoral research in Belgium. So he stayed as a postdoc, and then finally he left us in the end of 2020 to become a professor in 2021 in Nanjing. And you know, I'm very glad to say that Hua has been very successful in Belgium. So here, for instance, he got the award of the BIRD, which is the very well-known Association of Industrial Companies on R&D in Belgium. He got the award from them, and at the right side, you see that this award was delivered to him, actually by the King of Belgium in 2018. So just to express my knowledge and my strong appreciation for all the achievements of Hua while he was for 10 years in Ghent with us. So what are the research disciplines? And so, you know, in Ghent, we are working on nucleic acids as new biotherapeutics to cure modern, of course, numerous diseases in the future. So we deal with mRNA, DNA delivery, siRNA delivery. So our group in Ghent is a multidisciplinary group. And so we are doing nanotechnology, biophysics, cell biology, biophysics as well, preclinical and clinical work. And so actually in the field of nanotechnology, for the polymers, for the materials, and also in our biophysics work, when it comes to the use of light, we actually collaborate with Qiaobo and with this Hua and with the people in the joint lab in Nanjing. So the current activity, so what we do, of course, you know, in normal times, I make summer visits to Nanjing to give lectures. And so I did it for a couple of times. So we have joint PhD students. So our next student will defend his PhD in Ghent and in Nanjing in 2021, in the end of 2021. We applied for joint research applications like at FWO. And of course, we also make joint research publications. And I'm very glad, for instance, to tell you the most recent ones. So here, we have a very recent publication from our joint lab where we actually designed the kind of polymer films which can be activated by light and which we are investigate for the delivery of nucleic acids into the surface of the eye. Yeah? So it's a recently published work. And also very last week, actually, so we had our publication, Nature Nanotechnology, on a new type of material. So these are electrospun fibers, which we actually can activate by light for the transfection of all kinds of cells, especially for T cells in the context of T cell therapy. So here we have kind of joint activity on material design for biological purposes. And I'm very glad as well. So here we have also Ran Hu was the first author of the paper. This was the end of his stay in Belgium, I would say. And here, so we also have actually, based upon that work, we have also installed recently a spin-off at Ghent University. So we have been very, very collaborative and very productive, in our relationship with our Chinese friends. So which are the added values? Very short. So of course, I try to support the research at NFU. I try to expose, of course, the students to English lectures, which is important as well, because of course, English remains a quite big challenge for many students in China. Of course, I try to bring some financial support to the group, visibility in China. I try to use my network. I'm editor of the GCR, which is the main journal in the field. And of course, I try to use my network to offer it to my colleagues in China as well, and to facilitate, their work, their scientific work as well. Of course, we offer them to send students to EU, and of course, we keep our relations and friendship. And what is the added value from my side, the way I feel it, is that I have a gate to China. And so it's very important to stress that, because China is a big country. So for Belgian and European scientists, it remains a big country and sometimes very difficult to find your way. So for me, it's a kind of gate to China. So Xiaobo and Hu are helping with me. So to build a Chinese network, of course, also to apply for dedicated funding from EU with Chinese partners. And as you can see here, so most important for me is that I enjoy the friendship, as you can see at the bottom of this slide. This is something I really want to stress, that I enjoy this friendship, this collaboration with Xiaobo, with Hua, with the students very much there. Of course, that's real life. And so it's about traveling, about visiting each other, but it's also about enjoying each other. And we have already enjoyed a lot of activities, a lot of pleasant moments together, both in Belgium as well as in China. I'm very grateful that strong collaboration that is very productive. And I would like to thank the whole group there in Nanjing, let's say, for this nice opportunities we get from them. And I hope I can visit you very soon. Thank you.
Yeah, it's really a nice introduction to the laboratory. To my understanding, the joint laboratory is kind of a son of the Professor John Ball. I think there's also a good collaboration examples between the University of Ghent and the 1943 University. I have a question here. What were the challenges or difficulties in the very beginning when you really want to build up such kind of joint laboratory? Did you have any difficulties or challenge to overcome?
So maybe Xiaobo, you can, and Hua, you can first comment on that. I can then continue.
Yes, okay. Stefan, thank you very much. My voice is clear?
Yes.
That's good. Thank you very much, first of all, for your presentation. It taught me a lot. Because I didn't speak English for quite a long time. So if I can speak in Chinese, I can show more about what I'm feeling now. But now the English is not good than before. So anyway, I'm quite happy and impressed by your presentation. Everything we did before is a friendship. This is I and Hua field. Friendship is the base for everything, of course, including research cooperation. I think one word is quite impressed me during my public defense. In 2011, December 2nd, on my public defense, you told me we are friends, and even I'm your son in China, and you can call me you can call me anywhere, anytime, you know, without anything about the evening, early morning, everything like that. This is quite impressed. Of course, this topic, today's topic of the seminar is about cooperation, for including joint lab between Chinese University and Belgium University, of course, two partners. What we did now is quite successful, we think. As you showed in your presentation, the official start is from 2 years ago, 2.5 years ago. Our president of Nanjing Forestry University visited Uganda in 2019, yes. And Stefan, you invited our president to have a lunch together with them. And my president is quite appreciate for that. After visiting to Europe, he told us we should get more cooperation between Uganda and Nanjing Forestry University. After half year, I think in December, November. In November, the vice rector of Uganda have a visit to China, and then you visit together with her in China, and then we signed the official contract for our joint lab. But of course, the joint lab is an open platform for everything, for student exchange, research, cooperation, everything what we can do. Of course, including funding obligation between China and Belgium, Flanders. Of course, it's quite useful for us. Also, we did a lot based on joint lab. We also do some cooperation with a cosmetic company, as you showed in your presentation, Oshman. It's a very famous cosmetic company in China. The time is quite limited. In summer, friendship is everything. It's very important because we have many colleagues in our university. Not everyone can have such kind of a joint lab. Our joint lab is quite successful between me, Hua, and Stephen, even Kevin together. Friendship is quite important. It's the most important thing. What do we think? Hua, maybe Hua can say something.
Yeah, we still have 2 minutes. Yeah, maybe Hua,
2 minutes.
Hua to give some comments on the outlook. What is the future of the joint laboratory? What's your plan, and what are your expectations?
So actually, you know, so before, I think it's before the pandemic, we have already, so like Stefan, there we visit China for at least 1 time or even 2 times per year. But now we only have the online meeting. Yes. So maybe after the pandemic, and we will start again. So it's we will invite Stefan to come to China again. And the most important, I would next say, probably one of our students will go to Stefan's group to start the PhD research. So that's it. We will keep. So it means we will keep this kind of relationship with Stefan's group, and then we will make it more and more intensive cooperation. So in the future. So that is my comment. I think it's very. So it's very, how to say, as Chao say, it's a friendship. It's very important, and the most important. I think it's most important is we trust each other, and I you know when you do the cooperation, So you should trust your friends. You should trust your collaborator. Otherwise, there's many conflicts between each other. So
okay.
Let's say, and also one most important was to the people who you would like to build the cooperation with your promoter or with your friends in Belgium or in other countries. So I think that's the most important you should keep in your mind. So
thank you, Renhua. For the sake of time, I'm afraid that we have to conclude here. Thank you all, Steph, Chabo, and Zhang.
Yeah, we are quite happy. Yeah, we made many old friends today.
Yeah. I
we
really
knew
appreciate
each other. Yes, 10 years ago in Ghent.
Yeah, I really appreciate for your acceptance of the invitation and the preparation of the interview. And I hope you will continue your success with the joint laboratory and develop more talents for both countries. So to close this session, I would thank also the invited speakers again and appreciate all the participants for your attendance. I hope you will further enjoy the next sessions on green building, which is hosted by my colleague, Dr. Wan Ling. Thanks a lot. Bye-bye.
Bye.
Bye-bye.
Ladies and gentlemen, distinguished guests from China and Belgium. After 2 days of intensive agenda and the third China-Belgium Science and Technology Symposium is coming to an end, and we are about to enter the closing session. During the closing session, Mr. Ma Xiangjun, the president of the Association of Chinese Enterprises in Belgium and Luxembourg, and Mr. Jiang Yang, the secretary of Science and Technology Department from the Chinese Embassy of People's Republic of China in the Kingdom of Belgium, will present us with a summary of the conference and its outlook. Please first welcome Mr. Ma Xiangjun. The floor is yours.
Dear friends, ladies and gentlemen, dear organizers, ACPB and CBTC and other organizers, very nice to see you here. It was such a pleasure for me to join this big event. In the past 2 days, I just witnessed a very interesting symposium. And congratulations to the successful symposium. ACBL was established in 2004. As of its establishment, we aim at the development of this economy and trade between China and Belgium in order to improve the good partnership. So this year is the 50th anniversary of China-Belgium diplomatic relationship. During the past 50 years, the two countries made lots of achievements during the cooperation. According to the status of the Belgian Foreign Trade Agency, in 2020, China and Belgium trade in goods reaches amount 26.7 billion euro. It's a 11.7% increase compared to previous years, among which Belgium export to China reaches amount 8.54 billion euro, and import from China 18.16 billion euro. For Belgium, China is a top 9 export market and a top 5 import market, which is also the largest trade partner in Asia. By end of the first half a year of this year, the China and Belgium trade reaches amount of 14.9 billion euro, increase 16.6% compared to the previous year, which shows a very good outlook. According to 2020 statistics, so bullet of China's outward foreign direct investments, the China make investment to Europe around $12.6 billion US, increased 20% compared to last year. During the COVID, the Belgium to China investment still increased 210%, which broke the record for the past. By end of 2019, ACBL members make investment of 1.4 billion euro in Belgium. ACBL make a survey recently regarding the situation in Chinese enterprises in Belgium. The survey shows half of the interviewed enterprises are optimistic on local market. Majority of Chinese companies are confident about further developments in local market, even under COVID pandemic. The China-Europe relationship encounters big challenge, but over 50% interviewed companies expressed the satisfaction on current business performance and intention to active involvement in local market. Over 70% interviewed companies are cautiously optimistic about the coming 5 years. 70.4% interviewed companies intend to invest for expanding overseas business. Ladies and gentlemen, China and Belgium has cooperation in science and technology for over 40 years. Since 2015, China and Belgium signed several cooperation agreements and documents regarding science and technology, which push forward the cooperation. A batch of ACBL members are devoted to technology, research and development in Belgium, such as Huawei, ZTE, Dahua Technology, BGI, HIK, Vision, the China Unicom, China Telecom, Cloud, Accentric. Yesterday, we are very glad to hear the exchange between the companies, institutions between China and Belgium. And we are very glad to see the new progress of CBTC. We sincerely hope CBTC will open successfully soon and become a new hub for high technology. The third China-Belgium Science and Technology Exchange Symposium focused on bioscience, AI, climate change, green building, and economic cooperation. By sharing successful experience and outstanding achievements during the past 50 years, the symposium provides new opportunities for both Chinese companies and Belgian companies to achieve mutual complementarity in the interest of our common development. At present, a new round of science and technology revolution is in rapid development, which plays more and more important roles in economic growth. Emerging technology such as IT and AI expanding rapidly. China is opening up to the world into a high level. The 14th five-year plan of China decided innovation driven as further development's direction. In face of the big challenge for mankind's survival caused by climate change, China will make great efforts to achieve peak carbon dioxide emission by 2030 and carbon neutrality by 2060. Recently, China released related official documents which specified the intention to enhance the cooperation with the other countries on green technology, green equipment, green service, and green infrastructure construction. Belgium is very strong in technology innovation. According to 2019 Global Competitiveness Report released by the World Economic Forum, Belgium ranks top 22 among 141 economic entities. Innovation capacity ranks top 17th. Belgium is also among the leading countries in the field of nanotechnology, chemical engineering, microelectronics, biopharmacy, etc. The Belgium has a scientific and technology advantage, while China market has a large commercial opportunities and a great demand. This creates favorable conditions for mutual cooperation. ACBL encouraged the enterprises to enhance the cooperation with local governments, local companies in information communication technology, biopharmacy, environmental protection technology, high-tech materials, high-end manufacturing, etc. ACBL will make efforts to create good environments and provide assistance for members and youth, and promotes the commercialization of science and technological achievements. For more contributions to the improvements of China-Belgium bilateral economic and trade partnership. Thank you very much. Thank you. Bye-bye.
Thank you, President Ma. And next, I would like to invite the Secretary Jiang Yang to give his summary of the symposium. Hello, Jiang Yang, please.
各位嘉宾、女士们、先生们、朋友们，我是驻比利时使馆江阳，很高兴与大家在线上见面。Ladies and gentlemen, distinguished guests from China and Belgium, I am Jiang Yang from Embassy of China. It's a great pleasure to see you online. 很荣幸受邀在本次研讨会闭幕式上致辞。相聚的时光总是过得很快。过去的2天，来自中比两国的专家学者在生命科学、人工智能、气候变化、绿色建筑等多个重点领域进行了深入探讨。大会取得了圆满成功。当然，这离不开工作人员背后的辛苦付出和精心准备。在此，我仅代表驻比利时大使馆再次向旅比华人专业人士协会、中比科技园以及各协办单位表示衷心感谢。It's a great honor for me to be invited to address the closing ceremony of this symposium. Time flies when you are having fun. During the last 2 days, experts from China and Belgium have conducted in-depth discussions in various areas such as life sciences, artificial intelligence, climate change, and green building, etc. The symposium has been a great success, which would not have been possible without the hard work and elaborate preparation by the organizers. So once again, I would like to express the gratitude to ACPB and CBTC and all the co-organizers on behalf of the Embassy of China in Belgium. Thank you. 今年我们迎来中比建交50周年，这是中比全方位友好合作伙伴关系承上启下、一网开来的重要一年。本次研讨会的前一天，刚刚召开了第2届中比科技创新对话。会上，中国科技部与瓦隆布鲁塞尔大区政府有关部门签署了新一期的研究与创新谅解备忘录。双方将在生物技术和健康、信息通讯、微电子、航空航天、可循环材料、环境和绿色技术等多个领域持续开展联合项目研发、征集活动，其中很多方向都与本次研讨会高度契合。相信本次研讨会的举办必将对促进中比各领域科技交流合作产生积极的推动作用。希望大家以此次会议为契机，加强沟通交流，深化团结合作，取得更多的务实合作成果。中国驻比利时大使馆将一如既往地为中比科技交流与合作提供支撑和服务，支持两国在开放式创新中实现互利共赢。This year, we celebrate the 50th anniversary of the establishment of diplomatic relations between China and Belgium, which is an important moment for the all-round friendly cooperative partnership between our two countries. This symposium comes just 1 day after the second China-Belgium Innovation Dialogue, where the Chinese Ministry of Science and Technology and the WBI signed a new MOU on research and innovation, which will see the two sides launch an ongoing call for joint projects in a number of areas, including biotechnology and health, ICT and microelectronics, aerospaces, recyclable materials, agriculture and food science, and green technologies. Many of these areas are highly relevant to this symposium. So I believe that this symposium will certainly have a positive impact on promoting the bilateral science and technology exchanges and cooperation. The Chinese embassy in Belgium will, as always, stand ready to provide services and support both countries in achieving mutual benefits and win-win results through open innovation and cooperation. 谢谢大家. Thank you for your attention.
Very good and comprehensive summaries from the two gentlemen. And like you both, and I'm very much looking forward to closer and deeper exchanges and collaborations in science and technology between the two countries, especially under these special moments of a 50 years anniversary. And I look forward to seeing you all next year's symposium, ideally in our brand new premises in Louvain-la-Neuve, the China-Belgium Technology Center. And I wish you all in good health and see you next year. Thank you. Yeah, very last session. Would you, everyone, please turn on your camera and we will have one big last shot of us all. Thank you. Please turn on your camera. With all the participants, we will have our one last photo. And it will be the end of this symposium. Ivan Song. We may smile, we may wave our hands, whatever we want to express our good expectations between the countries.